Thanks everyone for the time this morning, and I really appreciate the invitation from ProImmune and the opportunity to have this discussion with all the experts in the field. It's very exciting to see some old faces like Tim, Yongliang, who we met regularly during PAX 10 years ago. So very good to get together again. So today I'm going to talk about our integrated 3 layers pipeline for immunogenicity risk assessment based on our own in-house technology, experience, and also some human data. Just a quick view of DuoBio. We were founded in 2014, focused on the development of multi-domain-based, multi-specific biotherapeutic to address unmet medical need in the clinic. So when we say multi-domain, multi-specific, very complex proteins, that's why we consider immunogenicity is the key risk or liability for most of our pipeline candidates. At this moment, we were a subsidiary of Huadong Medicine, which is a publicly traded biopharma in China. We have multiple programs in the clinic, which I'll touch a little bit about in the next slide. And previously, we have worked with BioNTech and Merck. A quick snapshot of our pipeline. Our most advanced program is called DR10624 as a triagonist, 3 domains, triagonist against FGF21 receptor, glucagon receptor, and GLP-1 receptor. Very complicated protein, and it's now entering phase 3 trials. We also have multiple programs in phase 1 and phase 2 development stage. So when we think about biotherapeutics, obviously we think about immunogenicity. So on the left panel, I'm showing some FDA-approved biotherapeutics, and on the right panel, you can see some red ribbons that represents the predicted T-cell epitopes, and we can see those predicted T-cell epitopes are just everywhere throughout the protein. But some are real problems, some are not. So even when we look at their clinical ADA rate, they varies one from the other. That tells us immunogenicity is a very complicated problem. The predicted T-cell epitopes, most of the time they can correlate with immunogenicity or ADA response, but sometimes they do not. So the take-home message would be therapeutics. Therapeutic proteins can trigger anti-drug antibodies, also known as ADAs. ADA may impact safety, for instance, hypersensitivity, pharmacokinetics, the PK profile, efficacy, if we develop neutralizing anti-drug antibody, and it can be a major cause of clinical failure. Especially the bococizumab, the PCSK9 map, which team knows everything, right? So that's a famous example when we think about why immunogenicity is so important for biotherapeutic development. And again, it's multifactorial. I know this picture may have been used many times during this workshop, but when we look at the immunogenicity, it can be the sequence. That's what we are doing with in silico development, but it can also be glycosylation. For instance, the syntaximab, although it's a chimeric antibody, but it's my favorite chimeric antibody. It has a murine sequence, but it has relatively low immune response in humans. The most frequent immune response actually comes from its glycan that triggered IgE response. So if we use a different expression host, the EGFR antibody is purely safe, purely great. So that's the problem of glycosylation. Obviously, formulation contaminates, application route, dose, even for dose, which I'm going to touch a little bit at the end of my presentation. Most of the time, those can be a bigger factor compared with a sequence. So when we look at the immunogenicity from a biotech perspective, we think the biggest challenge is no single model reliably predict human immunogenicity. And we have so many limitations. For instance, the species differences, animal studies was human, and also even if we use human use the Vitro or Vitro human PBMC assay, again, it's an incomplete immune system representation in Vitro. Besides, if we look at in silico prediction, a lot of time we get over prediction. So, and regarding the solution, we really need a multi-layered integrated strategy. So that's what we have developed in Duo. We have these actually four layers of immunogenicity assessment. So the first layer, what we call in silico sequence-based prediction, and at this time I will only focus on the T-cell epitopes. So what we're looking at is computational screening for MHC class 2 binding T-cell epitopes. The strategy is obviously high throughput, low cost, and it's very applicable for early stage development when we have so many hundreds or even thousands of these sequences. Of course, we have our own algorithms, and each company has its own language model to do the in silico prediction. But in general, it's very easy to operate and easy to scale up for high throughput screening. And the outcome is we can use this method to guide our initial protein engineering, especially for the immunization, which Yongliang and I have been working even back in college. And the limitation is, first, it's only predictive, it's not validation. Besides, when we look at in silico prediction, it's only sequence-based, or most of the time linear sequence-based. So it's not functional prediction or assessment, and most of the time it will overestimate the risk. Even if we look at a human antibody, that can be predict T-cell epitopes, but if we align that with a human repertory sequence, it's perfectly fine. The second layer is what we call in vitro ex vivo human PBMC assays. It can be MAPS, ELISpot, or simple CD8 T-cell depleted proliferation assays. The strength would be, first, it's human-derived cells, so it's a human, not animal, not mice, rodent, or monkeys. And it has this functional process where the therapeutic protein is actually being processed by antigen-presenting cells. So we can have a mechanistic insights into the T-cell activation. Obviously, the outcome's good. We can have functional human immune response. We are very diverse HLA types if we have a big, good, reliable PBMC donor shop. But we still have some limitations. The first one would be donor variability. Especially as a biotech company, we have limited resource to find the PBMC donors, so the quality can be different. Besides, it's not a systematic administration. It's only in petri dish, so we don't have this immunogenicity assessment where a systematic context. So the third layer we have implemented is the AD profiling and PK analysis in non-human primates, and most of the time it's cynomolgus monkeys. The strength is the best available proxy for human systematic response, although it's not human, but a lot of times it's very close to human. Especially if we look at vaccine research and vaccine development, most of the time non-human primates can represent human immune repertoire or immune response. And I still remember a famous MAPS publication 10 years ago. It analyzed the correlation between immune response in non-human primates and human trials, and it's almost like 50% chance that you're going to get a right correlation. So still, it's the best available technology these days. However, and when we look at the outcome, it's also quite different with the in silico prediction and the PBMC assays. We can have the opportunity to look at the correlation or the relationship between PK profiles and immunogenicity. And if we choose the right monkey models, for instance, a monkey model of obesity or diabetes, we can even get the opportunity to analyze the PD markers, the relationship between PD markers and immunogenicity. So it's a very good systematic model prior to human. And of course, there's some limitations. We still have it's a typo, it's the monkeys variability. Different monkeys are different. Besides, it's very expensive. So at the last layer, it's human trials, human studies. That's the moment of truth. That's when we have the real idea or good idea about how the therapeutic protein perform in human subjects. We can get a real immune response and impact on the PK profiling or some even PD markers like the PCSK9 map we just talked about. And the limitation is what it is. Once we enter in human trials, there's just no way to come back. Most of the time, we won't have the opportunity to just re-engineer protein and get it back to the human studies. If it failed, it fails. So at Dual Biologics, we use these first 3 layers as our integrated pipeline to provide in-house integrated risk assessment and candidate ranking. So when we look at the first 2 layers, we need to think about the immune response in the context of first antigen presenting. We have the peptide that can be recognized by the class 2 MHC alias. Then it can lead to the T cell activation, activated T cell, CD4 T cell epitope, which can trigger the translation from native and naive B cell to memory B cells and plasma B cells, which can eventually generate or produce anti-drug antibodies. So our first layer, we look at the computational screening for MHC class 2 binding T cell epitopes. And the second layer, we look at the T cell activation and proliferation assays, where we can use different methods to profile the T cell activation process. So here's some example we have for the in silico prediction. We have built up our in-house algorithms to look at the potential T cell epitopes in any sequence. So for instance, we have 3 different therapeutic candidates, and all of them are heavy chain only antibodies or VHHs. So the first one we consider it at high risk. And by the way, the green line here represents the predicted CD1 region. The blue line represents the predicted CD2 region, CDR2 region, sorry. And the red one represents the predicted CDR3 region. So for instance, the first candidate we have, it looks generally fine within the CDR region. However, it has significant risk in the constant region, in the framework region, which we can potentially introduce mutations to de-immunize or to de-risk this candidate. The second one, we call it a moderate risk antibody lead. And it looks generally fine on CDR1, CDR2, and CDR3 region. Actually, they are the same family of antibodies against the same target, but have different framework regions. And by introducing some mutations, we can de-risk this candidate. And for the last one, we can completely eliminate the potential risk by introducing multiple mutations. That's what we call a low-risk candidate. That's purely based on our in-house in silico analysis. And when we look at our in-house human PBMC assay that was designed and developed by Zheng Xin, who is also here in the conference today. So we have the CD8 depleted T cell proliferation assay. The PBMC samples are purchased from local vendors. And when we look at its gene background, mostly Chinese donors, not surprising, and that is HLA, DRB1 alias. And when we look at the potential limitations, it would be first, most of our PBMC are from healthy volunteers. We don't have the luxury to collect PBMCs from patients or from representative patient population. And also, since we have limited resource, most of the PBMC comes from very random HLA alias. And also, since we only have the local vendors, it has very limited racial diversities. However, it's still good technology for us to analyze the immunogenicity risk of our therapeutic candidates. And last layer would be the non-human primates. And it's very standard preclinical PK and tox studies in cynomolgus monkeys. The blood samples are normally analyzed for ADA and PK profiles. It's been very expensive these days. Last year it was completely different, but for this year it's very expensive due to the shortage of monkeys, and that's due to the booming of in vivo CAR T and the cells gene therapy in the world. So here I would like to provide some case studies to see how these 3 layers of risk assessment look in real world, especially in human studies. So the first one would be a VHH antibody, which would predict to be low risk based on our in-house in silico algorithm. But when we look at the ex vivo human PBMC assays, the positive rate jumps to 31%. So we conclude it's a moderate risk. And when we move forward to the animal studies in non-human primates, the ADA positive rate also changes. You can see the majority of the monkeys have developed anti-drug antibodies to the drug to the protein. So in this case, we consider it as a high immunogenicity risk. However, we still decided to carry its lead candidate to clinical studies due to various reasons. And in our human studies, what we found is only 20% of the subjects have developed anti-drug antibodies. So I will call it a moderate immunogenicity risk. And even we have the real title of anti-drug antibodies, it has limited impact on the PK profilings. So when we look at this example, I will say it's still a general, reliable risk prediction, because based on the results from the first 3 layers prediction, I will still consider moderate risk for the therapeutic proteins, and we can still translate it into clinical studies. And in the end, what we get is perfectly fine. It works generally well tolerable in human subjects. The second case study or second example I can share with us is different from the previous one. It's not antibody, it's actually a human protein, or I will call it engineered human protein. So we can't use the in silico prediction because it's a human protein, and we introduce five amino acid mutations. And among those 5 mutations, 4 are considered hydrophilic or charged, and that's our favorite amino acid substitution when we perform deimmunization. And that's because when we look at the MHC curve, it's actually hydrophilic. So when we have hydrophobic. So when we have hydrophilic amino acid substitution, it's generally safe. So I don't need to worry, or I don't need to care about any in silico prediction. And when we look at ex vivo human PBMC assay, it's fine. It's 15% donors develop the positive response, so it's still generally low risk. However, when we look at the non-human primates studies, it's extremely high. Almost every monkey develop anti-drug antibody against a protein. In fact, we even need to negotiate or communicate with the regulatory agencies about how we design the long-term toxicology study, because in non-human primates, eventually the ADA titer is going to be too high. All the drugs are going to be cleared away, so it's not representative. So it has been quite challenging for us to communicate with the regulatory agencies about the immunogenicity risk. However, based on the ex vivo PBMC assays, plus it's a human protein with favorable amino acid mutations, we still decide to carry it out to human studies. And you can see it has excellent immunogenicity profile in humans, almost no ADAs. In our phase 1b/2a study, which in this case, 41 subjects received the therapeutic protein weekly for 12 weeks, repeated dose study. Only 5% developed ADA. And in our phase 2 study, where we have almost 60 subjects receive the therapeutic protein weekly for 12 weeks, again, repeated dose study, only 3.4% develop ADA. So that's another perfect example. If we only look at the immune response in non-human primates, we may just drop the program because the risk is too high. However, if we take both PBMC assay into consideration plus our knowledge or know-how about how to de-immunize the protein, it can correlate perfectly with our human experience. And we have performed the both in Silico prediction and PBMC assays in-house. We get the opportunity to compare the results side by side. So for this table, I'm showing the in Silico prediction and PBMC assay for 12 proteins. Some of them are considered as medium or low in our in silico prediction. One was considered as high. And when we look at its correlation in PBMC positive rate, I would say it's fine, it's okay, it's not bad, it's not 100% correlation, but it's not bad. However, if we look at another 12 proteins we perform in-house, the correlations seem to be very poor. So that's another example to emphasize the complexity of immunogenicity prediction in the preclinical settings. And when we look at the results, some of our thought would be the limitation of in-house PBMC assays. First, we have limited PBMC samples, small scale, low ratio and HLA diversity, and also the suppliers of our PBMC assays. And also, since we are not a dedicated vendor for PBMC assays, the assay handling can be a potential problem. Also, the technical details, experience, and know-how. Besides, we do not have a dedicated team for quality assurance or quality control for PBMC assays. So that's why some thoughts to improve the quality of the reproductivity of the PBMC assays would be work with professional vendors, and obviously ProImmune is a good choice for these studies. And in this slide, I'm showing the comparison of our in vitro PBMC assays with our in vivo ADA studies in non-human primates. Again, it's a selective of proteins which we have tested in both PBMC assays and non-human primates. I think, except for 2 proteins, in generally, it looks good. It's not bad, the correlation is not bad. I will say it's much better than the correlation between Silico prediction and PBMC assays. So I think that's also another example to emphasize that we need to have different layers of immunogenicity prediction before we can draw any reasonable conclusion. So my first conclusion for our 3 layers immunogenicity risk assessment would be, first, no single layer is 100% predictive. The value lies in the convergence of data. How we interpret data, how we look at data is the key. And the most impact, for example, would be our engineered human proteins. It behave differently in different models, and eventually our knowledge and understanding of the immunogenicity actually help us to make a right decision. Also, this 3 layers integrated platform is very good for our candidate ranking. So we can integrate the scores from all 3 layers to allow us to choose the superior lead compared with a single assay. And for the strength and limitation, we have talked about a lot, so that's a quick summary. For the in silico, the throughput is ultra high, but it's only theoretical, and a lot of time just over-predict. However, it's still based on human. For our second tier, the in vitro, it has medium throughput. It's based on the cellular response, so better than the theoretical prediction. And also we're using human cells. And for our third tier, we move to the in vivo stage, where we have very low throughput, extremely low throughput. And most of the time, we can only test one or no more than two candidates. But it gives us systematic review of how the immune response look like for any particular candidate. And unlike the first two layers, we're using non-human primates, so it's quite different from humans from the immune genetics background perspective. And when we get back to the complexity of immunogenicity of proteins, I'd like to touch very quickly on the dose. Because when we look at the sequence, no matter it's in silico prediction or PBMC assays, what we're looking at is the sequence, it's the amino acid composition. However, a lot of times that can be a lower impact compared with the dose. So in this case, I'm using a famous example of Botox. It's a bacterial protein, so it's highly immunogenic. If we use in silico prediction, it's everywhere. The T cell epitope is just everywhere. High density of T cell epitopes. And if we use PBMC assays, very strong response. If I remember, it's like 60 or 80% response rate from PBMC assays. However, its immunogenic profile is quite different in different doses levels. For instance, if we're treating globular lines or front lines, we only use 20 units, and based on the historical rate, only 1% develop anti-drug antibody. But does that mean it's a safe and stealth protein? It's not. So if we look at cervical dystonia, each treatment needs 200 units. And if we look at the historical data, the ADA rate jumps from less than 1% to actually 14 to 20%. And around 10% of the subjects or patients will develop neutralizing antibody. Eventually, will compromise their treatment efficacy. So that's another example of the complexity of immunogenicity. Same sequence, it will have almost the same evaluation results in our 3 layers of prediction, but in clinical translation or in the real world, they can have completely different immune response. But do we have a solution? Potentially. So that's a very interesting project Yongliang and I worked during Dartmouth College, where we have this publication on synthetic biology, where we describe a high throughput screening and engineering platform to deimmunize the light chain of Botox. And we also have a US patent application pending, and that's Yongliang here. That's how he looked like during college. So my final conclusion would be the integrated 3 layers pipeline provides us structural risk assessment. So it's a very useful tool or very useful platform technology to have in-house. And we look at immunogenicity risk mitigation. Using these 3 layers of assessment, we can have a better candidate ranking. We can identify risks early to prevent costly late-stage failures. Besides, it also improved our clinical success probability. And the take-home message, again, single assays are insufficient. 3-layer integration can help us to make better decisions. And the early de-risking is critical for biologic success. And I guess the final word from me is, if you have the luxury, perhaps you can work with a professional vendor like ProImmune, and that can be really helpful. Thank you. Happy to take any question.
Thank you, Dr. Fang. I have a question. What is the result of your PBMC assay, and how many doses you use to test immunogenicity? And what is the criteria to categorize the risk of immunogenicity?
Sure. So Zhenxing, can you answer the question? He is the expert to answer your questions.
Okay, thank you for your question. For the first question, we use the CIFSE labeled the CT4 positive cells as in the PBMC assay. So after stimulation with the protein for 7 days, we can observe the we can observe the proliferation of the CD4 T cells. So meanwhile, we use the KLH as a positive protein and the medium as a negative control. So we compare the signal between the protein and the medium, and we can decide which protein is positive in the assay.
Another way to answer the questions, for the negative control, obviously DMSO. For the positive control, is always KLH. And if you have the luxury, you can sprinkle some famous antibodies, for instance, adalimumab or Vivo, that's considered as a high immune response and antibodies, so you can put that as your internal control for the PBMC assay.
Great talk, Dr. Fang. So I saw the 2 case study that you were sharing on that 3-layer, which I particularly like that kind of thought process of the risk in terms of understanding at every top of in silico, for in vitro, followed by doing the monkey studies. So just looking at the data there that you have, it seems that most, or if not all of the monkeys, they actually produce the ADA. So my first question to this is that, is it necessary to actually perform the monkey study to understand the immunogenicity, looking at the ADA response?
Yes, I guess the answer is absolutely yes, because for biologics, especially in China, the regulatory agencies rely heavily on the toxicology profiles in monkeys. Without knowing the immunogenicity in monkeys, it's impossible to draw any comprehensive conclusion from toxicology studies. For instance, the monkey may look perfectly safe, perfectly tolerable. Is it because the toxicology of the risk of the lead candidate is low, or because the monkey develop neutralizing anti-drug antibodies that completely swipe out the therapeutic protein from the monkeys? You never know.
Right.
So the immune response studies in monkeys is critical. It's actually key for any biologics.
Right, that makes sense. And second part to my question on this exact slide. So I was thinking, if let's say if we were to switch the scenario whereby the in vitro PBMC assay were to show a medium to high risk level of, let's say, 40%, let's say, and then the animal study ADA, you would be looking at a low response. Then what would your decision in terms of taking this molecule forward?
Right. So I have to look at the exact sequence to say based on the PBMC assay and in silico prediction, it seems to be moderate or high. However, let's say that's high. For moderate, we can probably still carry it forward, but for a high risk profile from both in silico prediction and PBMC assay, while low risk from non-human primates, I will have to look at the potential indication, whether we are going for oncology indication or whether we are going for non-oncology indication. Because for oncology indication, most of the time, the immunogenicity is not that critical, except for T-cell activation or T-cell engager. It's a different story. But for most immune response, no, I should not say that. If it's not an immune agonist, for instance, 4-1BB that actually failed because immunogenicity, so I should correct myself. If it's not a T-cell activation therapy for oncology or non-oncology indication, it should be fine. But if we are working on a T-cell activation therapeutic candidate for oncology indication or for any non-oncology indication, I will be very careful.
Right.
So it really depends on the clinical indication, depends on the mechanism of action in humans.
Right, thank you so much.
Yeah, thanks for the presentation. Actually, I have the same question from Simango. So based on the two cases you shared, could we conclude that NHP actually poorly correlate with the clinical outcome?
I won't say that because first it's a limited case study, so it's quite challenging to draw any statistically reliable conclusion. Besides, we have seen quite different response in non-human primates and humans, but we also have seen similar immune response in monkeys and humans in our own hands. So I don't think I can draw that conclusion.
Thank you.
The three-layered assessment what you have performed?
Sorry, what's the question?
Is the three-layered assessment what you have performed, is it something in alignment with the NMPA requirements?
No, I think for NMPA requirements only the non-human primates. There's the drafted guidelines for so-called preclinical assessment. I remember in the draft they talk about T-cell proliferation assays, especially for, let's say, T-cell engagers or immunomodulate therapeutics, but it's not a requirement. In fact, most companies in China, they do not perform these two layers of assessment. But because I have this unique background, so I have implemented this pipeline in-house.
Yeah, is it on? Yeah, so on the Botox data, that's quite interesting with the sort of dose dependency. Sometimes, I guess, once you go to a high dose, you may induce tolerance.
Right.
And I guess with Botox, I'm assuming because it's such an incredibly active molecule, what the 20 units are is such a low amount.
Yeah, 20 units is like 7 to 9 picogram.
Yeah, so probably the germinal center density of that presentation isn't enough to differentiate an antibody response. Yeah.
Yeah, but that's for asymmetric application, but for therapeutic application, 200 units is not even the highest dose I've seen. I've seen up to 1,000 units for some special indications.
Yeah, yeah. So the potency may sort of, then sort of link a little bit the sort of level of antigen that you can get presented in lymph nodes and so on.
Yeah, exactly.
Yeah.